bacteria structure and reproduction. So, in this lesson we are discuss about the bacteria structure and their importance and their size and shape and reproduction. So, before going to the bacteria, we are discuss a few lines about the cell. So, what is the cell? Cell is the basic structure and living unit. So, in that cell all the metabolic activities such as respiration and photosynthesis and digestion and excretion. So, these all metabolic activities are controlled by the cell. So, in that cell first discovered by the scientist name of Robert Hooke in the year of 1635, and Robert Hooke wrote a book, the name is Micrographia. In 1673, Leeuwenhoek is a Dutch scientist first examined the microorganisms and named them animalcules because of their dazzling movement. Then next in the year of 1839, cell theory, so that is proposed by two scientists, Sleeden and Swan. So, according to the cell theory, all the living organisms which are made up of pre-existing cells. So, all the living organisms are made up of pre-existing cells. All the living organisms, so which are composed of one or more cells, and so these cells which are developed from pre-existing cells. So, according to the cell theory, the cell is the basic structure and functional unit of living things. The next, cell lineage theory. So, that is proposed by Rudolf Virchow in the year of 1855. So, according to the Rudolf Virchow, cells arises from pre-existing cells by the cell division, mitosis or meiosis. Now, the cells are classified mainly two types. One is the eukaryotic cell and the second one prokaryotic cell. So, in eukaryotic cell, there are two type of cells are present. One is the animal cell and the second one plant cell. So, in eukaryotic cell, based on the presence and absence of cell wall, so these are classified into two categories. One is the animal cell. So, in that animal cell, cell wall that is absent, as in plant cell, cell wall that is present on the outer surface of the cell membrane. So, it means presence and absence of cell wall. So, these cells are classified into two types, animal cell and plant cell. And some are the differences between the animal cell and plant cell. So, in that mainly chloroplast that is present in plant cell and that chloroplast is absent in animal cell. In plant cell, a large vacuole that is present in the plant cell, and vacuoles either present or absent. If it is present, they are small in size in animal cells. Then next, prokaryotic cell. When compared to eukaryotic cell, prokaryotic cell absence of some cell organelles such as mitochondria, Golgi bodies, endoplasmic reticulum and a well developed nucleus. So, these cell organelles and these are membrane bound organelles. So, these are absent in prokaryotic cell. When compared to eukaryotic cell, such membrane bound organelles are absent in eukaryotic cell. Then next, flagella. So, if the flagella are present or absent, if the flagella is present, that is motile in nature. If the flagella are absent, that is non-motile. So, based on the presence and absence of flagella, cells, so especially prokaryotic cells, so these are classified into two categories. One is the non-motile and motile. So, as in non-motile cell, flagella are absent, as in motile cell, flagella are present. So, then next, the main difference between prokaryotic and eukaryotic, one is the nucleus. So, well developed nucleus that is present in eukaryotic cell and well developed membrane bound nucleus. So, that is absent in prokaryotic cell. So, it means this nucleus lacks membrane structure, only nucleoid that is act as a major role in prokaryotic cell, while in eukaryotic cell membrane bound nucleus that plays a major role in eukaryotic cell. Then next ribosomes. So, ribosomes are present in eukaryotic and prokaryotic cells. So, it means both cells having ribosomes. In that eukaryotic cells having 80S type of ribosomes and prokaryotic cells having 70S type of ribosomes. In that 70S type of ribosomes, 50S and 30S two subunits are present in prokaryotic cell. So, it means 70S type of ribosomes, they can classify it into two categories. One is the 50S type and the second one 30S type. So, these ribosomes scattered in the cytoplasm of prokaryotic cell. Either these are individually arrangement or either these are formed in group. So, if the ribosomes are formed in group, that is called polyribosomes. So, these ribosomes main site for the protein synthesis. So, in eukaryotic cell and prokaryotic cell, protein synthesis takes place on the ribosomal. In eukaryotic cell and prokaryotic cell, in both cells protein synthesis takes place on the ribosome. Let us discuss bacteria. So, bacteria, it is a plural word, bacterium. So, that is a singular word. So, bacterial members are unicellular microscopic organisms. Before the invention of microscope, we do not know about the bacteria. So, bacteria first discovered by the scientist name of Antony van Leeuwenhoek and named them animalcules because of their dazzling movement. The next, Ehrenberg, the bacteria name which is given by the scientist name of Christian Ehrenberg. The next, the importance, Louis Pasteur given the role of importance of bacteria and wrote a book, that is germ theory. So, germ theory, the book wrote by Louis Pasteur. Distribution of the bacteria. So, bacteria distribution, so it is cosmopolitan in distribution. So, it means they can present in soil, air and water, and a few members of bacteria, they can live in high temperatures such as 100 to 120 degrees temperature. So, they can live high temperature also. Such type of bacteria are called thermophilic bacteria. So, it means thermophilic bacteria, they can live high temperatures such as 100 to 120 degrees centigrade, and some of the bacteria present deeply in the oceans and soils, and a few members of bacteria parasites and few members are saprophytes, and a few member of bacteria mutually symbiotic association with plants. For example, Rhizobium bacteria, it is present in the leguminaceae plants. So, in leguminaceae plants, nitrogen fixation by the help of Rhizobium bacteria. So, molecular nitrogen that is present in atmosphere, that is mainly N2, dinitrogen form. So, in this dinitrogen form of nitrogen in atmosphere, the molecular nitrogen that is present mainly in the form of dinitrogen. So, these dinitrogen plants cannot utilize for their metabolic activities. So, these dinitrogen convert into nitrates and ammonia with the help of bacteria. So, bacteria size. So, bacteria size ranges between the 0.5 micrometer to 2 micrometer in width, the next 2 micrometers to 5 micrometer in length, and the largest bacteria, that is Epilopium phycelsoni. So, in recently one of the another bacteria that is observed, and that name is Theomargarita namibiensis. So, it means largest bacteria, that is Theomargarita namibiensis. So, it is approximately 750 micrometers in length, and the smallest bacteria, that is Pasteurella pestis. It is considered as the smallest bacteria. Shapes of bacteria. So, bacteria having different shapes based on their cell wall composition. So, it means cell wall gives definite shape to the bacteria. So, there are different forms are present. One is the cocci, bacillus, spirochetes, vibrios and spirilla. So, these are different shapes of bacteria, different types of bacteria. One is the Streptococcus pneumonia and Clostridium tetani, Trypanosoma, and that is spirillum bacteria, Vibrio cholera, it is vibrio shaped bacteria, and Salmonella typhi, that is rod shaped bacteria, and Helicobacterium pylori, Clostridium botulicum, and Staphylococcus aureus. So, they are given different forms of bacteria. So, let us discuss one by one. So, first one, coccus bacteria. Coccus, it is a singular word, cocci, that is a plural word. So, in coccus bacteria, what is coccus bacteria? Round or spherical shaped bacteria are called coccus bacteria. So, in this coccus bacteria, number of cells present in it. So, these coccus bacteria classified into different types. In that monococcus, diplococcus, streptococcus, tetracoccus, staphylococcus and sarcina. So, these are different types of coccus bacteria. In that first one, monococcus, a single spherical shaped bacteria, that is called monococcus. Now, the second one, diplococcus, a pair of spherical shaped bacteria, so that is called diplococcus. Next, tetracoccus, 4 spherical shaped bacteria, that is called tetracoccus. So, it means tetracoccus bacteria, in that they are forming in tetrads. So, tetra, it means 4. Now, the next one, streptococcus, as in streptococcus bacteria, more than 4 bacteria, so which are fused and forming a chain like form, that is called streptococcus bacteria. Next, staphylococcus bacteria, a number of bacteria which are fused in irregular forms, so that is called staphylococcus. So, streptococcus and staphylococcus, so both are having a number of bacteria, but streptococcus having a particular shape. It means streptococcus bacteria, a chain like bacteria. In staphylococcus bacteria, it is a irregular shape. And the last one, sarcina. In sarcina, 8 spherical shaped bacteria which are fused and forming a cuboidal shape. So, that is called sarcina. So, these are different types of coccus bacteria. Now, the next one, bacilli. Bacilli, it is a plural word, bacillus, that is a singular word. So, rod shaped bacteria, that is called bacilli. So, rod shaped bacteria. So, based on the number of cells present in it, so these bacillus bacteria classified into different types. In that first one, monobacillus and diplobacillus. Then next, streptobacillus. First, monobacillus. Mono, it means only single, a single rod shaped bacteria, that is called monobacillus. And diplobacillus, a pair of rod shaped bacteria, that is called diplobacillus. The next, streptobacillus, a number of rod shaped bacteria, are joined one to another and forming a chain like form, that is called streptobacillus. So in that one of the another bacterium that is present, polysaid bacterium. So it means a number of bacteria, rod shaped bacteria are arranged irregular form, that is called polysaids. Next, spirillum. So spirillum, spiral shaped or ribbon shaped bacteria, so that is called spirilla. And vibrio, comma shaped bacteria are called vibrios. And the next one, spirochetes. Some of the spirilla bacteria are flexible and it is referred to as spirochetes. So based on the position of cell wall and type of cell wall present in the cells, so these bacteria classified into different shapes. And one of the bacteria is present, that is called pleomorphic bacteria. So pleomorphic bacteria, it is a irregular shape bacteria. It means based on the type of environment and nutrients available, so such bacteria change their shape and structure. Such bacteria are called pleomorphic bacteria, and pleomorphic bacteria shows a process that is called pleomorphism. Azotobacter bacteria, it is a best example to the pleomorphic bacteria. Based on the type of cell wall present in it, bacteria are classified into two types. One is the gram positive bacteria, second one gram negative bacteria. You see in this image, gram positive bacteria, so it is purple in color, and gram negative bacteria red in color. So what is the gram positive, gram negative? So presence and absence of cell wall material, so it is classified into gram positive and gram negative. So it means entire the bacterial cell wall that is made up of two components, one is the thicoic acid and the second one muramic acid. So as in gram positive bacteria, thicoic acid and muramic acid both are present, while in gram negative bacteria, thicoic acid is absent. So it means presence and absence of thicoic acid, gram positive and gram negative. So based on the presence and absence of cell wall material, so these are classified into two types, gram positive and gram negative. So in gram positive bacteria, cell wall material, thicoic acid and muramic acid, so both are present in gram positive bacteria, while in gram negative bacteria, thicoic acid is absent. So it means only muramic acid act as a major role in cell wall. So Christian Gram developed a gram staining procedure. So based on the gram staining technique, so bacteria we can easily identify gram positive and gram negative. So this is the gram staining procedure. So in this gram staining procedure, first we have to prepare bacterial culture, and this bacterial cultural slide treated with different type of reagents. In that, first of all we are used to crystal violet, then next it is treated with iodine, then next 95% of alcohol or acetone, and finally the bacterial stained slide that is treated with safranin. And finally, so this bacterial cultural slide observed under the microscope, we have to find out two types of bacteria, one is the purple color and the second one red color. So if the purple color that is indicates gram positive bacteria, red color it is appear that indicates gram negative. Ultra structure of bacteria. So let us discuss the structure of bacteria. So bacterial members are unicellular microscopic organisms. So we can observe only under the electron microscope. So, bacteria observed under the electron microscope, different parts which are clearly visible. In that, we are discuss main important organelles and their role. First of all, cell wall. Bacteria surrounded by a thick layer that is called cell wall. Cell wall gives a definite shape to the cell and protects from the osmotic regulation and turgidity. So, on this cell wall, different type of outer growths which are developed. On the bacterial cell wall, different outer growths are formed. In that, one is the pili or fimbriae, flagella, next sex pili. So, these are the outer growths which are formed on the bacterial cell wall. First, we have to discuss flagella. So, flagella, so mostly flagella are present in the bacillus type of bacteria. So, it means bacillus bacteria consist of a number of flagella and the number is 1 to many flagella are present on the outer surface of the cell wall. So, with the help of flagella, bacteria moves from the one place to another place. If flagella are present, such bacteria are called motile. If the flagella are absent, such bacteria are called non-motile. And these flagella made up of a protein that is called flagellin. And the next one, pili or fimbriae. On the bacterial cell wall, a number of pili or fimbriae are formed. When compared to flagella, pili it is short and straight and rigid in nature. So, with the help of pili or fimbriae, bacteria attachment on the solid surfaces. Next, sex pili. So, in between the pili, one of the largest hair-like structure is present, that is called sex pili. So, during the bacterial conjugation, genetic material that is exchanged from donor bacteria to receptor bacteria with the help of sex pili. So, bacterial cell wall outer growths are mainly flagella, pili and sex pili. Next, protoplasm. Below the cell wall, the fluid material is present. So, entire the homogeneous fluid material, so that is called protoplasm. In protoplasm, cell membrane, ribosomes and nucleoid, cytoplasm. So, these are the main organelles scattered in the protoplasm. So, in that first one, cell membrane. So, cell membrane it is a living membranous layer which is present below the cell wall. So, it is a living membranous layer and it is made up of polysaccharides, lipids and a few amount of proteins. So, in cell membrane, lipids and proteins, so both are present. So, that is why cell membrane is lipoproteinaceous. So, it means lipids are present and proteins. So, both are associated in the cell membrane. So, that is why cell membrane is lipoproteinaceous. Next, below the cell membrane, homogeneous fluid material, so which is scattered. So, that is called cytoplasm. So, in that cytoplasm, a number of ribosomes are scattered. If the ribosomes are individually arranged or so they are formed in group. If the ribosomes are formed in group, that is called polyribosome or polysomes. So, cell membrane invaginates and infolds and forming a specialized structure that is called mesosome. So, these mesosomes are help in binary fission. And next, nucleoid. So, bacterial cell, it is a prokaryotic cell. So, it means true nucleus is absent, only nuclear material is present, that is called nucleoid. So, in bacterial protoplasm associated with genetic material, so that is called nucleoid. So, nucleoid having a single chromosome and non-histone proteins. So, it means histone proteins are absent in their DNA. In bacteria, another genetic material that is present, so that is called plasmid. And reserve food materials stored in the form of glycogen. So, based on the type of flagella present on the outer surface of the cell wall, bacteria can be classified into different categories. So, in that first one, atrichous bacteria. So, bacteria do not having flagella on their surface, so that is called atrichous. So, it means atrichous bacteria, non-motile bacteria. Second one, monotrichous bacteria. If the flagella only one flagellum, that is present only one end, that bacteria is considered as a monotrichous. Amphitrichous bacteria, a pair of flagella are present on their both ends, that is called amphitrichous bacteria. And peritrichous bacteria, a number of flagella developed on the outer surface, that is called peritrichous. And lopotrichous, a tuft of flagella developed on their one end, that is called lopotrichous. And cephalotrichous bacteria, a number of bacteria developed on their both ends, so that bacteria is called cephalotrichous bacteria. So, it means based on the number of flagella present on the cell wall, so bacteria can be classified into different types. Reproduction. So, bacteria reproduces asexual and sexual methods. In that, asexual method, one is the binary fission, and sexually reproduces by conjugation, transformation and transduction. First one, asexual reproduction. Bacteria reproduces asexually by the method of binary fission. So, during the favorable conditions, bacteria reproduces asexually by binary fission. So, during favorable conditions appear, bacteria reproduces by binary fission. So, what is binary fission? So, in the binary fission, parental cell divides and produces two daughter cells. So, by a process that is called binary fission. So, during the binary fission, genetic material, that is nucleoid, present inside the cell membrane. During the binary fission, so in binary fission, cell elongates after the formation of genetic material. Now, the center of the cell, so mesosomes which are present on the cell membrane, now the mesosomes increases centripetally and dividing equal distribution of protoplasm in two daughter cells. After the distribution of protoplasm, cell wall divides and separates two daughter cells. So, this is binary fission. Then next, sexual reproduction. So, bacteria sexually reproduces by three methods: conjugation, transformation and transduction. So, first, conjugation. So, during the conjugation process, some of the bacteria having genetic material plasmid and some bacteria do not having plasmid. If the plasmid contain bacteria, that is called donor bacteria. Absence of plasmid, such bacteria are called receptor bacteria. So, during the conjugation process, bacteria such as donor bacteria and receptor bacteria comes to close to each other. In that time, genetic material such as plasmid exchange from the donor bacteria to receptor bacteria with the help of pilus or pili. So it means donor bacteria transfers the genetic material to receptor bacteria with the help of pili. So in sexual reproduction, transformation and transduction, in a transformation, the genetic material DNA enters into the bacteria from the environment. So that is called transformation. So this transformation process first discovered Lederberg and Tatum in Streptococcus pneumonia. During the transduction, the genetic material enters into the bacteria through the bacteriophages. So that is called transduction. Transduction, the genetic material exchange from the bacteriophage to bacteria. By this process, first discovered by the scientist Lederberg and Ginder.